Hi guys, today we will be looking at a quick revision on the topic of transport across cell membranes. We will cover simple diffusion, facilitated diffusion, active transport, osmosis and co-transport. So here I've drawn the phospholipid bilayer. So the structure of the cell membrane, if you remember, it's made of the phospholipids. They're arranged in the way when the phosphate and glycerol are facing outside and the fatty acids are facing inside. We also have the channel proteins within the membrane. We've got the carrier proteins, and also if you remember properly with the structure of the cell membrane, we can find the cholesterol which will provide fluidity. We also have the glycolipids, which you can find attached to the phospholipids, and we have glycoproteins, which are on the surface of the proteins embedded within the membrane. Those glycolipids and glycoproteins got the function in the cell recognition. You've done that topic with the structure of the cell membrane. So, the aim of today's quick session is to recap on different types of the transport. So, we will start with the simple diffusion. Simple diffusion is a movement of small lipid soluble molecules down the concentration gradient. Down the diffusion gradient. So what does that mean? It happens from the area of higher concentration to the area of lower concentration. Example of the molecule that can diffuse through the phospholipid bilayer is, for instance, oxygen. So, simple diffusion is a movement of small lipid-soluble molecules, so those are the properties of the molecules, down the concentration gradient through the phospholipid bilayer. And that was simple diffusion. There is the other type of the diffusion, which is also a passive transport, which happens down the concentration gradient from the higher to lower concentration. This type of diffusion, it's called facilitated diffusion. Facilitated diffusion will move larger molecules and non-lipid soluble. This type of diffusion also will transport the molecules down concentration gradient but this time it's not going to happen directly through the phospholipid bilayer because the molecules that we are transporting through facilitated diffusion are larger and non-lipid soluble. They will be transported using proteins. Proteins involved in the transport through the facilitated diffusion could be channel protein or carrier protein. So, one more time, facilitated diffusion, it's a movement of larger non-lipid soluble molecules down the concentration gradient, so from higher to lower concentration, using channel or carrier proteins. Another type of the transport that you should be aware of, it's the active transport. Active transport then is the movement of the ions of the charged molecules against the concentration gradient. This process, as it says, it's active, so requires ATP. And because we're moving charged molecules, it needs to happen through the protein. However, the protein responsible for the active transport, it's the carrier protein. So active transport, as we've said, it's the movement of molecules against concentration gradient. Hence, they will move from the area of lower concentration to area of higher concentration through the carrier proteins using ATP. Another type of the transport is the osmosis of water. So osmosis is the process where we need to use different terminology. Osmosis, so we use different slide for the osmosis. Osmosis is the movement of water from higher water potential to the area of lower water potential. The idea of higher and lower, so the comparison statement, it's really important on your exam. So that process, it's called osmosis. And it also, what's also important to mention, it's the movement of water. So osmosis, it's the movement of water from the area of higher water potential to the area of lower water potential. Many students don't understand what that means, so I'm going to show you in a really simple way. Okay, so imagine here is the level of water. And here we've got the substances dissolved in the water, let's say glucose. On the other hand, I'm going to show you the lower water potential. So this is the level of water. And this is then glucose. So as you can see in a really simple way, the water potential is the measure of water. So that means we've got more water but less solids dissolved in there. In the higher water potential, lower water potential means otherwise in a really simple way, less water and more things dissolved in there. The final, and that is also a passive process. So same as it was simple diffusion and facilitated diffusion, the osmosis is also a passive process. Final process of the transport across the membrane that you need to be aware of, it's co-transport. Co-transport is a movement of two different molecules. So you should be familiar with the movement of glucose and sodium ions, which you've discussed in the topics, for instance, absorption of glucose. So for the co-transport, I'm going to use a cell, which is the epithelial cell. Epithelial cell has many adaptations for the co-transport. For instance, we've got the increased surface area, we've got a large number of the mitochondria. Remember, mitochondria is the site of aerobic respiration, which produces ATP. As you can see, ATP will be produced in the epithelial cell, hence we will be talking here at some point of the active transport as well. Another adaptation of the epithelial cell is the fact that they've got many proteins, carrier proteins. So I'm going to draw quite a few. The reason why I'm using different colors is, of course, that proteins got the specific 3D tertiary structure, so we know that each protein will be responsible for the transport of different molecules. Another adaptation is the fact that it's close by to the blood capillary, so provides the short transportation pathway. And here I'm going to draw the lumen of the epithelial cell. Okay, in the lumen we can find sodium ions and glucose, which we want to obviously transport into the blood capillary for the absorption. So the easiest way of understanding co-transport is starting with the movement of sodium ions. Sodium ions are actively transported into the blood. So this is the active transport. We just had a look at the active transport a second ago, so you should remember the active transport, it's happening against concentration gradient, requires ATP and carrier proteins. So we can check if we've got all that in place in here now for co-transport. Yes, the green one, we've got the carrier protein. This transport requires energy, requires ATP, because it's taking place against concentration gradient. So step 1, we had the active transport of sodium ions from the epithelial cell into the blood capillary by active transport. The direction is important, so we need to say from epithelial cell into the blood. Obviously, that now maintains diffusion gradient for sodium ions. Because if we transported sodium ions out of the epithelial cell, the concentration of sodium ions in the epithelial cell now decrease. That gives fantastic conditions for the sodium ions to now diffuse from the lumen into the epithelial cell. However, by doing so, they're taking with them glucose. This is the evidence of the co-transport. Two different molecules are transported. So that is step number 3. Movement of the sodium ions and glucose from the lumen into the epithelial cell. That now increases the concentration of the sodium ions, but also increases the concentration of glucose. This again maintains the concentration gradient of glucose, increases the glucose concentration inside of the epithelial cell. And glucose is classified as the larger molecule. So coming back to our transports, we now need to think how the glucose is going to be transported. It's not going to be transported via through the active transport, because it's going to go from higher to lower concentration gradient, so down the concentration gradient. So it's going to be a passive process. So we can pick between simple diffusion or facilitated diffusion. However, as we've mentioned, glucose is a larger molecule. Being larger, it requires the help from the channel or carrier protein. And this is why I've drawn here the red carrier protein, which allows glucose to now be transported and absorbed into the blood. So we've got here number 5, a facilitated diffusion of glucose carrier protein down the concentration gradient from the epithelial cell into the blood. So remember, directions are really important. To start our process one more time, we've started with the active transport of the sodium ions from the epithelial cell into the blood capillary, and this process, it's an active process, hence requires carrier protein and ATP going against the concentration gradient. Taking sodium ions away from the epithelial cell lowers the concentration, hence maintains the diffusion gradient for sodium ions. Hence sodium ions now with the glucose will be transported through the facilitated diffusion from the lumen into the epithelial cell. That movement increases the concentration of glucose inside of the epithelial cell, maintaining the diffusion gradient for the glucose to now move through the facilitated diffusion from the epithelial cell into the blood capillary using the carrier protein. So those are the main transports that you need to be ready to recall for your exam: simple diffusion, facilitated diffusion, active transport, osmosis, and co-transport. Don't forget to think about the factors affecting each of the transports. So main things to consider here will be, for instance, temperature, will be, for instance, the number of the available proteins, the saturation of the proteins, the maintenance of the concentration gradient, and so on. So remember, practice is important. So get on with some past paper questions now. So that's everything on the transport.